Good morning, students. So, we are discussing a new chapter, photosynthesis. In that, early experiments. What we are discussing, that is Joseph Priestley candle jar experiment we have discussed in the last class. Next, the other experiments are Jan engine horse. So, the next experiment, which is Jan engine horse. So in Jan engine horse, he proved that. So for every experiment, what their conclusions? So what does he prove? That sunlight produce oxygen. So sunlight produced oxygen. So what he stated, that the green parts, the aquatic plants, in aquatic plants, the green parts in aquatic plants, the green parts, they produce, in the presence of sunlight, they produce small bubbles. In sunlight, they produce small bubbles. That is in the presence of sunlight. So in the absence, whereas in the dark, or in the absence of the sunlight, these bubbles are absent, which are absent in the dark. That means the green parts which undergo photosynthesis, those green parts, they release oxygen in the form of bubbles. So that indicates that the aquatic plants, they release the water bubbles, oxygen in the form of water bubbles. That was stated by Jan engine horse in the year 1730. So this was the proposal the Jan engine horse was observed. Okay. Next, Julius von Sack. So what he stated, that the production of glucose So here, if you see the glucose production, that is the chloroplast. What he stated, chloroplast consists of chlorophyll. Pigment chlorophyll, which undergo photosynthesis, and produce glucose, which is stored as starch. So that is what generally Julius von Sack stated. the year 1854. So production of glucose is the main thing. Here sunlight produced oxygen. So glucose is stored. Glucose is produced during the photosynthesis and stored in the form of starch. That is his proposal. And next, T.W. Engelmann. The next scientist, T.W. Engelmann. So what he stated, that the action and absorption spectrum, he proposes the action and absorption spectrum, action spectrum and absorption spectrum. Action spectrum and absorption spectrum. So he has performed the experiment, that is with algae, cladophora and aerobic bacteria. The prism, he has passed the light through the prism and he placed in the water, he placed the algae and bacteria. He placed the algae and bacteria. So here the sunlight falls on this. So when the light falls on the prism, it diffracts, and then the different wavelengths of the light, say suppose with DrERK. So these wavelengths of the light passes through the medium containing the algae and bacteria. Okay, listen carefully. When the light is passed through the prism, it diffracts, and then that is passed through the medium containing the algae and aerobic bacteria. Which bacteria? Aerobic bacteria. So here what happens? When always photosynthesis occurs at blue light and red light. Photosynthesis occurs at blue light and red light. So wherever the blue light is present, blue light wavelength is present and red light wavelength is present. So there if you see the blue and red wavelengths of the light, the blue and red wavelengths, blue wavelength is nothing but at 470 to 500 nanometers, note down, whereas red is 650 to 760 nanometers. At that particular wavelength, the photosynthesis takes place. So the algae will absorb the light at blue and red light. So and they undergo photosynthesis at the area of the algae grows at region of blue and red. When the algae grows at a region of blue and red, there it undergo photosynthesis and evolves oxygen. So the bacteria which is growing in the medium, because in the other regions there is no oxygen, so the bacteria also moves to the region where algae is growing. Okay. So what you can confirm, as the air oxygen is present only, the bacteria is growing towards the nearer to the algae. So algae, where it is growing, wherever the wavelength is suitable for photosynthesis, that is nothing but blue region and red region. So this indicates that the spectrum of light which undergo photosynthesis only at a particular wavelength of the light, not in other regions of the light. It only produces the photosynthesis at a particular wavelength. Which wavelength? Blue light and red light. So other regions there is no growth of algae. So this is action spectrum. So action spectrum is at blue and red regions. Blue and red regions, that is action spectrum. Absorption, who is absorbing? Absorption spectrum is with, who is absorbing? Pigments. So pigments are nothing but chlorophyll A and chlorophyll B. Chlorophyll A and chlorophyll B is the absorption spectrum. Okay, right. So here this is what the proposals which are given by the T.W. Angelman. So action spectrum and absorption spectrum, that is light at a wavelength of blue and red regions. So ordinary photosynthesis occurs at regions of blue and red. And which pigment? The chlorophyll A and chlorophyll B. Okay, right. Next scientist, Cornelius van Niel. Cornelius van Niel. So here the purple oxygen is from water. Oxygen is evolved from water. This was the statement given by scientist Cornelius van Niel. Here what he stated, that the purple and green bacteria, so in the absence of water, if you see, there is no oxygen produced. That means in purple and green bacteria, the purple and green bacteria, the water is absent because these are photosynthetic bacteria. In purple and green photosynthetic bacteria, the water is absent, and in presence of that, the radioisotopic, the oxidizable compound, the oxidizable compound hydrogen sulfide is present. Hydrogen sulfide is present. So here if you see the H2A, carbon dioxide, CH2O, and water. So here if you see, there is no oxygen. The A indicates the sulfur or sulfate. Sulfur or sulfate is produced. If this is H2S, if this is hydrogen sulfide, oxidizable compound hydrogen sulfide, then the glucose CH2O, and whereas sulfur or sulfate is produced instead of oxygen. So this is proved by radioisotopic technique. So this is proved by radioisotopic technique. Radioisotopic technique. Okay, right. So this was the other experiment, early experiment by the Cornelius von Neil. So here what does he prove? That in green and purple sulfur bacteria, that is photosynthetic bacteria, where the water is absent, when the water is absent, there is absence of oxygen. That proves that the oxygen is produced only by water. Okay. So next is Rubin and Kamen. So here if you see, in 1941, Rubin and Kamen, he proved that using radioisotope O18, using radioisotope O18, he proved that oxygen is released by water. Proved that oxygen is released by water, or oxygen released by from water. So here he used the algae chlorella. By using algae chlorella, excuse me, by using algae chlorella, it proved that the oxygen is produced from the water. So here the O18, whatever the oxygen which is present, that oxygen which is say take the example of the H2O plus CO2. So here O is radiolabeled. So C6H12O6 plus here this oxygen which is released is the radioactive isotope, the oxygen, and along with that water. You don't see the radioactivity for the oxygen of water. The free oxygen only will have the radioactivity. That means the O oxygen of the water oxygen is radiolabeled. That is seen in the free nascent oxygen. So therefore only you can say that the oxygen is released from the water, but not from the carbon dioxide. So oxygen is from the water. So this is another experiment by Rubin and Kamen in the algae chlorella. They proved. So the algae chlorella, they proved that the oxygen is from water. Next, What is Hill's reaction? Hill's reaction, that is Robert Hill in 1937. Robert Hill in 1937. In 1937, what he stated that the chloroplast evolved oxygen in absence of carbon dioxide when illuminated in presence of a suitable electron acceptor. That means chloroplast evolved oxygen. The chloroplast evolved oxygen in absence of carbon dioxide. Because you see that the oxygen is from the water but not from the carbon dioxide. When illuminated in presence of suitable electron acceptor. In the presence of suitable electron acceptor, that is potassium ferricyanide. That is potassium ferricyanide. So Fe3 plus turns to Fe2 plus. Ferric to ferrous. Reduced. This is reduced during photolysis. So ferric to ferrous. So here, when you see the water in absence of carbon dioxide, means other than that you see the ferric compound, then it produces instead of carbon dioxide, a ferric compound is present, then it produces AH2, that is with whatever the carbohydrate product is there, that is, and oxygen. So here what is, when water is present, oxygen is released, but when carbon dioxide is absent, also you see oxygen is released. That means what is it indicates that oxygen is always produced from the water. Previously we told in the Rubin and Kamen, there if you see the water absent, then there is absence of oxygen. So this is also proved by the Hill's reaction. The other experiment which says the same thing, that oxygen is always evolved from the water. Okay, so these are the early experiments carried out by the different scientists. Okay, right. Next. Where does photosynthesis takes place? So where does photosynthesis takes place? So where does it takes place? As you already know that the chloroplast, you know the structure of the chloroplast. We have already studied in the cell the unit of life. So the chloroplast, if you see this chloroplast, it has pigments, the thylakoid membrane, pigments are present. So in chloroplast, pigments are present in thylakoid membrane. They are present in the thylakoid membrane. Pigments are present in the thylakoid membrane. That is, the carbon dioxide is fixed in the. So this is light reaction. So where you see light reaction occurs. What is light reaction? Let us see. Okay, we'll discuss what is light reaction later. Where carbon dioxide fixation occurs in stroma. This is dark reaction, which is dark reaction. So dark reaction is nothing but fixation of the carbon dioxide, whereas light reaction is nothing but to convert the light energy into chemical energy. Okay, right. So here if you see the photosynthesis, the pigments which are present, always the chlorophyll in the mesophyll cells, if you see, they are aligned. The chloroplasts are aligned in the mesophyll cells, perpendicular and parallel to each other. Where they are perpendicular, less light intensity. When they are having less light intensity, during less light intensity, the chloroplasts are arranged perpendicular to each other. They are arranged perpendicular to each other, perpendicular to the light. When there is a light intensity, low light intensities, the chloroplasts are arranged perpendicular, okay, to the light, perpendicular to light. Whereas if they are high light intensities, then they are arranged parallel to the light. Note down, when high light intensity is during high light intensity, the chloroplasts are arranged parallel to the light. Okay, right. So this is what we see in the case of the chloroplast. So when they are arranged in the mesophyll cells, so this is a mesophyll cell. So in the mesophyll cells, you know that there are a number of chloroplasts are arranged. So these are chloroplasts along the walls of the mesophyll cells. They are arranged. The chloroplasts are aligned along the walls. So to get the optimum light, to get the optimum amount of light, they are always arranged to the walls. So the light reaction, during the light reaction, light energy is converted to chemical energy. Let us see what happens in the light and what happens in the dark reaction briefly. So in the light reaction, the light energy is converted to chemical energy. Here, light energy is converted to chemical energy. That is what we see. Whereas in dark reaction, chemical energy is used for carbon dioxide fixation. The chemical energy is used for carbon dioxide fixation. Okay? Right. Next, the photosynthesis, splitting of water, occurs during the light reaction. Splitting of water, this is called photolysis, occurs during the light reaction. So water is splitted in the light reaction to release the electrons, protons and oxygen. To release electrons, protons and oxygen. So this is what we see in photolysis. So this will clearly explain how this splitting occurs, when it occurs exactly in the light reaction, that is cyclic and non-cyclic photophosphorylation. Okay, right. So there in detail you can come across this point again. So when the light is absorbed by the chlorophyll and transfers that in the form of electrons and protons to release the NAD, to reduce the NAD plus to NADPH. So that is all the big story for in the cyclic light reaction, where you call it as cyclic and non-cyclic. So photophosphorylation occurs. Photophosphorylation occurs in the light reaction. Photophosphorylation occurs in the light reaction to form the occurs and forms ATP and NADP. To form ATP and NADP. These are, I think you know, these ATPs and NADPs are energy rich molecules. So these are energy molecules. So cyclic and this is nothing but in cyclic and non-cyclic. So these are assimilatory power. So ATP and NADP are the assimilatory power. ATP and NADP are the assimilatory power, which this assimilatory power is used to fix the carbon dioxide in the dark reaction. So the carbon dioxide here in the dark reaction is fixed to form the glucose. So here glucose is formed by the fixation of the carbon dioxide. Okay, right. So it uses what? ATP and the assimilatory power ATP and NADPH. Assimilatory power ATP and NADPH is used in the glucose. Okay. So it occurs daytime but independent of light. So always dark reaction occurs in the daytime but independent of light. It is independent of light. So, whereas light reaction always occurs during the daytime. So, it is always independent of light. That means you don't utilize the light, but it is the product of the light, but depends on light reaction for their energy, and depends on light reaction, for energy. Okay, right. So this is with related to the some of the differences with the dark reaction and light reaction. Okay, right. So before going for the cyclic and non-cyclic photophosphorylation, what are these pigments? Let us study in detail about the pigments. So pigments are 5 to 8% of the total dry weight of chloroplasts. These are 5 to 8% of total 5 to 8% of the total dry weight of chloroplast. So the range is 5 to 8%. These are the substance that absorbs the visible light. Pigments absorbs the visible light. Pigments absorb the visible light. Next. So when you see this pigments, they are separated. The pigments can be separated by paper chromatography. You can clearly separate the pigments by using the technique called as paper chromatography. So when you separate this paper chromatography, the separation is by in order of if you see chlorophyll B, chlorophyll A, xanthophyll and carotene, they got separated. Okay. So these pigments, they are grouped under, the pigments are grouped under as chlorophylls, carotenoids, and phycobilins. Chlorophylls, carotenoids, phycobilins. Here if you see chlorophyll A and chlorophyll B comes under chlorophylls. Carotenoids are carotenes and xanthophyll. Whereas phycobilins are phycocyanin, phycoerythrin and yellow phycocyanin. Phycobilins are phycocyanin, phycoerythrin and yellow phycocyanin, which are present in blue-green algae and red algae. These are in blue-green algae and red algae. Blue-green algae and red algae. Okay, right. So these pigments, they give different colors. Like chlorophyll A, if you take bright or blue green, chlorophyll A which is bright or blue green color, whereas chlorophyll B, yellow green, chlorophyll B, yellow green, xanthophyll, yellow, carotenes, it is yellow orange. Carotene is yellow orange, that is yellow to orange. So this is what generally the different colors of the pigments. Okay, right. So in the next class we will see what is the wavelength of the light and what in which are colors, which light at which wavelength, that is nature of the light and all we will discuss in the next class. Okay, right. So thank you, see you in the next class.